Okay. Only 20 members are present. What about remaining? Okay. What are the last class we discussed? What is the bacteria? Yes. Anyone? What is the bacteria?
Bacteria is a single cell. We can see only electronic microscope only. It is a prokaryote, sir.
Okay.
It is a prokaryote and
Yes. What is the bacteria?
it has a known nucleus without a nuclear membrane, sir.
Bacteria are very small, microscopic, unicellular, and prokaryotic organism, right?
Yes.
The term bacteria was first used by the scientist name is. The term bacteria was first used by. The term bacteria first used by
Ehrenberg.
Yes. And the first observed by the bacteria first observed. The term used in Ehrenberg, the first bacteria observed by the scientist name is Anthony van
Leeuwenhoek.
Okay. What is the smallest bacterium? What is the biggest bacterium?
Mycoplasma gentium. Smallest bacterium. Largest Theomargarita namibiensis.
And Bagiota also. Okay. According to shape, how many types of bacteria?
Types.
How many types of bacteria according to shape? 3 types. First one is coccus or spherical,
Coccus.
or rod shaped, and spiral.
Spiral.
The coccus is divided the 6 type: mono, diplococcus, monococcus, streptococcus, staphylococcus, tetracoccus, and sarcina. And bacillus. The rod shaped bacteria is called bacillus, monobacillus, diplobacillus, and streptobacillus. And the third one is spiral or helical bacteria. These are the 2 types. Comma shaped bacteria, what we called Vibrio. Spirulum. Corkscrew shaped bacteria, it is called as spirulum. These are the in the previous class we discussed. Okay, coming to structure of bacterial cell. Ultra structure of bacterial cell. The ultra structure of bacterial cell, we see only electron microscope only, right? The structure of most of all the parts we can see only help of electron microscope. Ultra structure of bacterial cell, only we see electron microscope. Okay, already in the previous class, this is the diagram of bacterial ultra cell structure of bacteria. These are the 6 types of main important components of bacterial cell wall. The first one is slime layer. Already I told, already we finished in the slime layer also. What we call slime layer? What is the nature of slime layer? Yes.
Osmosis, exchange of food material.
No.
Water.
No. It is a gelatinous. It is a gelatinous layer or mucilaginous layer present on the outer surface of the. You see the diagram. Outer surface of the cell wall. This layer is called slime layer or capsule. It is made up of either polysaccharides or polypeptide or both also. It may depends upon the bacteria. Sometimes it's thin, sometimes it's thick. What is the function of slime layer? What is the function of capsule? It protects the bacterial cell from desiccation and antibodies. These are the function of slime layer. Next, cell wall. Next, cell wall. The cell wall is the beneath the capsule and external protoplasma membrane. The cell wall is present beneath. Beneath means below the capsule or below the slime layer, and external to the plasma membrane. External to the plasma membrane means
Above.
above. So the cell wall is present in the external to the plasma membrane or above the plasma membrane, below the capsule, right? The cell wall is the most important part of in bacterial cell. It gives what is the function of the cell wall? The cell wall function is it gives shape and protection to the bacterial cell. It is gives shape and protection to the bacterial cell. The cell wall is made up of polymer, peptoglycan, or mucopeptide or murine also. The murine, it is consist of a backbone of alternately arranged in the N-acetyl muramic acid in shortcutly NAM, capital NAM. NAM stands for N-acetyl muramic acid, and another one is NAG, NAG, N-acetyl glucosamine. These and the peptides are attached to the one side of the muramic acid. Each peptide consists of several amino acids, but alanine, glutamic acid, and diaminophilic acid are it is the very specific. So on your exams, you just write where it is present in the cell wall. It is the beneath lower side of the capsule and above the plasma membrane. The cell wall is present. It gives the function. The function is very, very important. The function of cell wall is shape, and it gives shape and protection to the bacterial cell wall. The cell wall is made up of a polymer, peptidoglycan, murine, or mucopeptide. Yes, these are the cell wall. Next, cytoplasmic membrane. Third one is
Plasmic membrane.
cytoplasmic membrane. You observe the diagram also. The cytoplasm is surrounded by a thin membrane. The cytoplasm is surrounded by a thin membrane. It is made up of proteins and phospholipids. It allows the entry and exit of small molecules. What the function of these cytoplasmic membrane? It allows the entry and exit of small molecules such as nutrients and waste products. These are the function of cytoplasmic membrane. The cell membrane acts as a differentially permeable membrane. It regulates osmosis. It contains several respiratory enzymes, the synthesis of capsule, the cell wall material. These are the cytoplasmic membrane. In this, the cytoplasmic membrane, it is mesosomes. The mesosomes also it's under cytoplasmic membrane. You see the diagram. You see the diagram. In some bacteria, not all bacteria, some bacteria, especially in the gram-positive bacteria, the plasma membrane invaginations. What is the meaning of invaginations? Yes. What is the meaning of invaginations?
It is the process of the separation of the cell
Which
into
allow the materials inside.
No.
Means which allows the
Invagination.
materials to which allows the materials inside the body.
which separates the cell into two fragments.
Simply,
It
the
it allows
invagina-
the it
just wait,
allows
just
the
wait.
materials to inside in the cell. Invasion means actually it
Simply the
allows the
invasion.
materials to enter into the cell.
Just wait. Just imagination means just the simple word is foldings.
Huh?
Foldings.
Okay, thanks.
Just wait. You
What is a
just
what
wait.
is a term, sir? Excuse me.
Yes. You see this diagram. Mesosomes. Invagination means folding. You see.
Inner
You
folds, I think.
yes. You see this diagram. You see this diagram. Especially in gram-positive bacteria, the plasma membrane invaginates into the cytoplasm. These
Plasma membrane.
once again I repeat, in some bacteria, especially in the gram-positive, plasma membrane invaginates into the cytoplasm.
Plasma membrane invaginate.
Please don't interrupt in the middle. You after completion of my class, you ask doubts. So these enfoldings are known as mesosomes. They were considered as the site of cell respiration. It is the function of mesosomes play role in the cross wall formation and DNA replication also. It plays role in the cross wall formation and DNA replication. Okay. Next, cytoplasm. Next one is cytoplasm. The bacterial cytoplasm is a complex mixture of, any idea? The cytoplasm contains, yes, anyone? Cytoplasm contains,
Cytoplasm contains nucleoid, plasmid, some ribosomes.
okay, the bacterial cytoplasm is a complex mixture. Mixture means number of components. The number of, what are the number of components? Carbohydrates, proteins, lipids, peptides, purines, pyrimidines, vitamins, coenzymes, etc. These all are it is a complex mixture. It is consist of cytoplasm. All the double membranous cell organelles such as endoplasmic reticulum and Golgi bodies are absent. Endoplasmic reticulum and Golgi bodies are absent the cytoplasm of bacteria. Although well organized chloroplast absent in bacteria. The chloroplast also absent in bacteria, but some photosynthetic bacteria have chloroplast in their cytoplasm. Yes, do you understand? Generally, the bacteria are absent in chloroplast, but the photosynthetic bacteria have, what is the examples of photosynthetic bacteria? Yes, Anabino, Nostoc. These are the photosynthetic bacteria. The photosynthetic bacteria means they prepare their own
Don't.
food material. They do not depend others. Okay. So what we call it is autotrophs. The nutrition of bacteria covers in the, what is the autotrophs, what is the heterotrophs? They cover. The photosynthetic bacteria have
Phototrophic.
yes. The
Autotrophic.
photosynthetic bacteria like Anabina, Nostoc, they have contain chloroplast, but most of the percentage of bacteria absent in chloroplast. Okay. What are the components? Complex mixture of cytoplasm, carbohydrates, proteins, lipids, peptides, purines, pyrimidines, vitamins, and coenzymes. The endoplasmic reticulum and cell organelles, Golgi bodies, are absent in the cytoplasm of bacteria, right? And the cytoplasm consists of some reserve food materials. The reserve food materials are fats, polysaccharides, phosphates. What are the reserve food material? Fats, polysaccharides, phosphates. The organic material stored in the form of glycogen and valutin granules and polyhydroxybutyrate. Polyhydroxybutyrate in shortcut PHB. PHB. And metaphosphate and polyphosphates are stored in the valutin granules. The polysaccharides are stored in the granules. These are the reserve food materials. And nuclear material. These are the very, very important. Nuclear material. What is the nuclear material?
Chromatin material.
What is the nuclear material? Nuclear material.
Plasmid.
Plasmid. What is the genetic material of bacteria?
RNA.
No.
It's DNA.
Yes, DNA. So
Virus.
two nucleic acids are there. So DNA is absent, automatically RNA is present. So you remember, students,
Sir.
the bacteria genetic, generally we will discuss in next class, the reproduction of bacteria. In reproduction of bacteria, there is no true sexual reproduction. How it is reproduction? Any idea? What is the reproduction of bacteria?
Conjugation, transmission,
Cell to cell contact.
transduction,
Just
cell.
wait. Cell to cell contact. So what happening in the cell to cell contact? The genetic material, one
Lex.
bacterium to another bacterium. The transfer of genetic material, one bacterium to another bacterium. That is the reproduction of bacteria. There is no true sexual reproduction in bacteria. The genetic material is DNA. So what about virus?
RNA is the genetic material.
In just wait. In virus contains DNA and RNA also genetic material. But suppose the virus contain as a DNA as a genetic material, RNA is absent. RNA as a genetic material, DNA is absent. But not at a time, the both are not present. Either DNA, either RNA. Okay, but bacteria's genetic material only DNA. The virus genetic material is DNA
or RNA.
RNA.
RNA. But both not present in the at a time. Suppose some virus present in DNA, RNA is absent. RNA present is DNA is absent. That is the difference between. What are the our genetic material?
DNA.
DNA.
Okay. The nucleic material of bacteria, it consists a single molecule of DNA. DNA is flexible, two-stranded, coiled, and circular. You know DNA structure. Already you studied in degree BSc level. What is the structure of DNA? What is the types
Double
of
helix.
DNA? Yes, the DNA is flexible, two-stranded, coiled, and circular. The bacterial DNA lacks the stone proteins. The very less amount of stone proteins are present in the bacterial DNA. It is called as nucleide. It is called as nucleide. The bacterial chromosome are genophore. It is not surrounded by a nuclear envelope. It appears in the amorphous lobar mass of fibrillary material. It lies freely in the cytoplasm. It replicates by semi-conservative method. So these are the nuclear material. Next one is plasmids. Next one is plasmids. In bacteria, extra chromosomal segments of DNA are present in the cytoplasm. What is the plasmids?
Extra chromosomal segment.
Extra chromosomal segments of DNA are present in the, where it is present? In the cytoplasm
Cytoplasm.
only. Cytoplasm only. It is called as
Plasmid.
plasmids. The term plasmid was first coined by Lederberg in the 1952. The term first used by the plasmid word first used by the scientist name is Lederberg,
Leidenberg.
1952. Some plasmids get integrated with the bacterial chromosome during the replication time. They are called episomes. Some plasmids get integrated with the bacterial chromosome during the replication time. They are called episomes. They are called episomes. By these episomes used in the scientist name is Jacob and Wallman in 1960. You know plasmids? What is the definition of plasmids? Simply extra chromosomal segments of DNA are called
Plasmid.
Plasmids.
plasmids. The plasmids, where it is present, the
Cytoplasm.
plasmids present in the cytoplasm.
Cytoplasm.
The term used in the plasmids, Lederberg,
Leidenberg.
1952. Some plasmids get integrated with the bacterial chromosome during the replication time. They are called episomes.
Episomes.
So next, ribosomes. The ribosomes also present in the cytoplasm. The ribosomes are sites of, what is the use of ribosomes? The ribosomes,
Protein synthesis.
yes, the ribosomes are the sites of protein synthesis in the bacterial cell in eukaryotic cells.
Okay.
The bacterial ribosomes are 70s type. It is consist two subunits, 50 and 30s.
30s.
50 and 50s and 30s.
30s.
Generally, ribosomes is how many types? Two types.
70s.
70s. Another one is
50. 50s.
50s. Only the bacterial ribosomes are 70s type. It is
Yes.
consist two subunits, 50s and
30s.
30s. And next one is flagella. Flagella. What is the flagella?
Flagella is for the locomotion of a bacteria.
Yes, flagella are a thin hair-like appendages. Hair-like appendages. They lack envelope. Each flagellum consists of 3 longitudinally coiled fibers. Each fiber is made up of protein called flagellin. Do you understand flagella? What is the structure of flagella? It is a thin hair-like appendages. They lack envelope. Each flagellum consists 3 longitudinally coiled fibers. Each fiber is made up of protein. Each fiber is made up of protein called flagellin. The structure of function, the structure of function of bacteria flagella have been discovered by the scientist name is Simon in 1978. Do you understand flagella? Yes. Yes, friends, please respond. The structures of flagella and functions described by the scientist name is Simon in the 1978.
1978.
These flagellas also in different types of flagellas are there. How many types of flagella is the 6 types of flagellas are there. 6 types of flagellas are there. Atrichus,
Monotrichus.
Monotrichus, Lopotrichus, Amphitrichus, Cephalotrichus, Peritrichus. Okay. The flagella. First of all, what is the flagella? Flagella is a hair-like appendages. It is thin structure, the lack of envelope. Each flagellum it is consists of 3 longitudinally coiled fibres. Each fibre is made up of a protein. The protein called is flagellin. The structure and functions of flagella who explained Simon et al. Okay, according to these scientists, how many types of flagellas? 6 types of flagellas are there: Atrichus, Monotrichus, Lopotrichus, Amphitrichus, Cephalotrichus, Peritrichus. The first one is Atrichus. Flagella are totally absent in this type of bacterium. Okay. What is the example is Naturella. Next one is Monotrichus. Next slide I show diagrams. So you see the diagrams. Monotrichus. The first you observe the first diagram. Atrichus. There is no flagella, right? All of you, you see the slide, the diagram slide.
Sir.
Yes.
Yes, sir.
Monotrichus. A single flagellum is present at one end of the cell. One end of the cell. It is called as Monotrichus. Example is Vibrio cholerae, Lopotrichus. A group of flagella is present at one end of the cell. A group of flagella at one end of the cell. Example is Spirulum volutans. And next one is Amphitrichus. One or more flagella occurs at both end of the cells. You see the diagram in the third one. One or more flagella occurs at the both ends of the cell. It is Autotrophs species. And Cephalotrichus. The Cephalotrichus not here. The Cephalotrichus, 2 or more flagella occurs at one end of the cells. The example is Pseudomonas. Right? Pseudomonas. Next, last one is Peritrichus. The cell surface is evenly surrounded by the several flagella. You see the 5th diagram. The flagellas it all over the body. The cell surface is evenly surrounded by the several flagella. Example is Bacillus species. Okay, these are the flagellas in the 6 types of flagellas are there. Okay, and examples also there. Next one is last one in the bacterial cell wall is Pili and Fimbriae.
Yeah.
Pili and Fimbriae. Many gram-negative bacteria possess or consist minute, fine, hair-like appendages extending towards from the surface of the cell wall. You see the diagram. Pili and Fimbriae. Many gram-negative bacteria possess minute, fine, hair-like appendages extended towards from the surface of the cell wall. These are called Pili or Fimbriae. See the diagram in the right side. Pilus or Fimbriae. This one. These are called Pili and Fimbriae. They are made up of special protein called pilin. The Fimbriae are Pili made up of a special protein. The protein name is pilin. They confer the property of this pili and fimbriae advances. The examples of pili, fimbriae, enterobacteriaceae and pseudomonas. Pseudomonas is the two examples of pili and fimbriae bacteria. The addition to pili, these are special type of pili known as. In addition to the pili, these are some special type of pili known as sex pili. The sex pili serve as passage for the entry of DNA from donor bacteria cell to the recipient bacterial cell. This will be discussed on the reproduction of bacteria. So that will what is the function of pili? What is the function of pili? We will discuss reproduction of bacteria. Okay, coming to
అందుకే పెనికిరవన్నీ నువ్వు నీట్ గా నేర్చుకోవాలి వాళ్ళు వెళ్ళి చేయాలి మన
please mute your phone. Some children's disturbance are there. Okay, so do you understand these are the structure of ultra cell structure of bacterial cell wall? Yes, students, do you understand this bacterial cell wall structure? What are the components of bacterial cell? Do you understand? Any doubts regarding this?
Sir,
Yes.
diagram, sir, pili and fimbriae diagram. Can you show me diagram, sir?
In the right side, in the corner, above filament, you see the pili, fimbriae, in brackets,
Okay.
fimbriae. Okay. These are the very important, the ultra cell structure of bacterial cell. Next, coming to nutrition of bacteria. What is the nutrition? What is the use of nutrition? The purpose of
Life.
nutrition.
Life.
Yes.
Produce energy.
Yes. ఇది న్యూట్రిషన్ ఎందుకమ్మా తెలుగులోనే చెప్పొచ్చు. ఏంది మీకు మీకు ఒక ఐడియా ఉండాలి కదా, ఎందుకు న్యూట్రిషన్ ఎందుకు అవసరం?
ఆహారం తీసుకోవడానికి.
Yes. ఆహారం తీసుకుంటే ఏమైతుంది? వాట్ హ్యాపెన్?
ఎనర్జీ
ఎనర్జీ
ఎనర్జీ.
Nutrition is for to live a life proper.
Okay. So it is common. You are daily taking tiffin, lunch, dinner, so many. So how to
Sir,
okay, for the disturbance. Yes, nutrition. How bacteria's the nutrition, how take the food, the bacteria's. Okay.
heterotrophic,
synthesis.
heterotrophic mode of nutrition.
In bacteria, the nutrition of bacteria's depending upon the source of availability of carbon.
Nitrogen.
The bacteria are differentiated into two groups. Yes, please silence. Generally, the human beings, some vegetarians, some non-vegetarians, right?
మేడం హెడ్ బాత్ చేసిన చెప్పాలి. ఇప్పుడు ఎందుకు పోనేదే ఈడేం చేయలేదా?
Please, the user Galaxy M202, please mute yourself.
Okay. Generally, the human beings is the food habits, the nutrition habits. How many types? Yes.
Autotrophs and heterotrophs.
No, no, no.
Heterotrophs.
I'm talking about human beings.
Human being. Okay.
Omnivorous. Herbivorous.
Yes. Tell me. Say loudly. Yes, girls. Sorry, students.
Heterotrophs. All human beings are heterotrophs.
What is the heterotroph?
Nutrition.
What is the heterotroph?
We depending plants and animals, sir.
Plants
They're
and
depending in
Yes.
Yes.
Saprophytic.
So, okay, girls. Sorry, sorry to say. Actually, I'm working. I'm working as a assistant professor in the Koti Women's College. So that is Women's College. Most of the I used in the girls. So don't. Most of the times the term used girls. So all of you, sorry. Okay, my dear friends, exactly right. Heterotrophs. We are the heterotrophs. Okay. We are the heterotrophs and some of the vegetarians, some of the non-vegetarians.
Yes.
How they define the bacteria? So the bacteria, the source is carbon. The availability of carbon. The carbon is where is it is? It is an atmosphere. Okay.
Yes, sir.
So in this, the major two groups is Galaxy M02. Okay. The major two types, autotrophs and heterotrophs. You know autotrophs. Autotrophs means
the plants. Autotrophs, the example plants that they're preparing own food.
ఎవరమ్మా అక్కడ ఎన్ని టైమ్స్ చెప్పినాం కొంచెం autotrophs. Who is children talking? Please mute yourself, please. I requested all of you. Just I question, just you answer, please. Otherwise, please mute yourself. So again, not listen my class, I think. Autotrophs. The autotrophs, the bacteria that can synthesize their own food materials, are called autotrophic bacteria. Yes. What is the meaning of autotrophs?
Bacteria
Which can
that can synthesize
their
their
Own
own
food
materials are
materials are called autotrophs.
autotrophic bacteria.
How they produce their food materials, utilize energy from either sunlight or chemical reaction for carrying out various metabolic activities.
వినిపించదా అది నేను రివర్స్ చేసేస్తున్నాను అందుకే
Iparamma, Galaxy M02s. I'm leaving the class, please. How many times? Number of times I muted that fellow, but he is unmute. So all of you unmute and your video also unmute. Just listen. There is no questions. So autotrophs, the bacteria that can synthesize their own food materials, are called autotrophs. They utilize energy from either sunlight or chemical reactions for carrying out various metabolic activities. What is the metabolic activity? Is anabolism. The metabolic activities is catabolism and anabolism. The photosynthesis is a anabolic process. What is the anabolic process? What is the anabolism? The anabolism, it is the complex molecules break down into simple molecules. Like the catabolic process, the simple molecules in the converted to the complex molecules. These are the metabolic parts. These topics are deeply studied in the photosynthesis and respiration part. So I'm going for that. These are the some metabolic activities are there. And what is the, before going to this, what is the photosynthesis? What is the photosynthesis? The photosynthesis is a, yes, it is the photosynthesis
is the
Yes,
process
tell me.
of the preparation of the food by using sunlight and water.
And the presence of the chloroplast.
Sunlight.
Okay,
Cross.
that is the photosynthesis. You know very well about photosynthesis. Okay. According to the source of energy, the bacteria, the autotrophs are before going to autotrophs. Okay, what is the heterotrophs?
Heterotrophs mean they can't prepare their own food
The
material.
heterotrophs bacteria are not able to synthesize their own food material. They obtain the food materials either from the living organisms or dead and decaying organisms or association with the living organisms like symbiotic, asymbiotic association. Okay, these are the heterotrophs. The heterotrophs bacteria we will discuss later. First of all, we discuss the autotrophs. According to the source of energy, the autotrophs are divided into following types. The first one is photosynthetic bacteria. Photosynthetic bacteria. These type of bacteria like green plants. The photosynthetic bacteria example is
Green sulfur bacteria.
yes. These bacteria like green plants possess the photosynthetic pigments and obtain the energy from the sunlight or and carbon from the atmospheric carbon dioxide. What is the photosynthetic bacteria? These bacteria, like green plants, possess the photosynthetic pigments. The plants, all the plants in the green, most of the plants in the plants of the plants in green color. Why it is green color?
Having chlorophyll.
Having
Chlorophyll.
the chloroplast contain chloroplast pigments. The chloroplast pigments, how many types of chloroplast pigments?
Three
You know,
types.
three types. What are the three types?
Cyano-
Carotenoids, xanthophylls.
Chlorophylls,
Carotenoids.
xanthophylls, and carotenoids.
Carotenoids.
The chlorophylls again, how many types?
Chlorophyll A, B, C, E.
6 types: chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E. Okay, chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, chlorophyll E. 5 types. And xanthophylls. How many types of xanthophylls? Yes, xanthophylls. A very basic question. In the 9th class also the chapter is there. The chloroplast pigments, xanthophylls. The xanthophylls also 3 types of xanthophylls, and carotenoids also 2 types of carotenoids. So, coming to our subject, the examples of photosynthetic bacteria. Green sulfur bacteria. These are the green sulfur bacteria. Is the green. These are the small and non-motile rod-shaped bacteria which are strictly anaerobic. What is the meaning of anaerobic and aerobic?
The reaction which involved in the absence of oxygen is called as anaerobic.
Yes, aerobic, the presence of oxygen, it occurs in the presence of the oxygen is aerobic. So, they utilize sulfate as a hydrogen donor. They are utilized CH4 as a hydrogen donor. The example is the green sulfur bacteria is Chlorobium, Heliobacteria. These are the green sulfur bacteria and purple sulfur bacteria. The purple sulfur bacteria, they utilize H2S, thiosulfate or sulfite as a hydrogen donor. Sulfite as a hydrogen donor. It is the purple sulfur bacteria. The example is Chromatium, Theospirillum, Theocytosis, Theocapsa. These are the examples of purple sulfur bacteria. Next, purple non-sulfur bacteria. They utilize organic compounds for carbon and molecular hydrogen as a hydrogen donor. The example is Rhodospirillum. These are the purple non-sulfur bacteria. Generally, the autotrophs, according to the source of energy, the bacteria are divided into two types. First one is photosynthetic bacteria. The photosynthetic bacteria, it is called as photoautotrophs. And second one is chemosynthetic bacteria, chemoautotrophs. How many types? Photoautotrophs, chemoautotrophs, or photosynthetic bacteria or chemosynthetic bacteria. The chemosynthetic bacteria. The chemoautotrophs. These bacteria are autotrophs as they use atmospheric carbon as a source of carbon and but derive energy from oxidation of inorganic substances. Oxidation of inorganic substance. What is the meaning of oxidation? Yes.
Removal of water.
What?
No. There is no water.
Oxidation,
Removal of
removal of
water is
Hydrogen.
hydrogen,
Hydrogen.
removal of hydrogen,
addition of oxygen, removal of
removal of
hydrogen
hydrogen.
is called as
Hydro.
oxidation. Oxidation. What is the reduction? What is the meaning of
Gaining
reduction?
of electron.
The reduction is it is opposite of oxidation.
Gaining of
Addition of hydrogen, removal of oxygen, it is called as reduction. So in this the chemosynthetic, it is the energy or carbon from the oxidation of inorganic substances depends on the type of inorganic substances. These are the three types. Chemoautotrophs are the three types. Sorry, four types. First one is hydrogen bacteria, second one is sulfur bacteria, and fourth one is iron bacteria, and fourth one is nitrifying bacteria. The first one is hydrogen bacteria. The oxidized hydrogen in the presence of oxygen, it is called as hydrogen bacteria. Example is Mycobacterium, Bacillus, and Pseudomonas saccharophila. Hydrogemonous bacteria are example of hydrogen bacteria. Next, sulfur bacteria. The sulfur bacteria obtain energy by oxidizing sulfur. The first one, hydrogen bacteria, it is oxidizing hydrogen. The sulfur bacteria is obtained by energy by oxidizing sulfur. So example is Bagiota. Bagiota oxidize the H2S and store the elemental sulfur as a granule in filaments. The example is Thiobacillus. Thiobacillus, example of sulfur bacteria. And next one is iron bacteria. The iron bacteria get their energy by oxidizing ferrous ions into ferric ions. The oxidizing ferrous ions to ferric ions, it is iron bacteria. Example is Ferrobacillus, Thiobacillus, and Gallionella and Nitrosomonas bacteria. Sorry, the iron bacteria oxidizing the ferrous ions to ferric acid. The example is Thiobacillus and Ferrobacillus. These are the examples of iron bacteria. And fourth one is nitrifying bacteria. The nitrifying bacteria are which oxidize ammonia to nitrates,
nitrites and
ammonia
nitrates.
to nitrites and nitrates. Nitrifying bacteria oxidize ammonia into nitrites and nitrates. Example is Nitrosomonas bacteria. The Nitrosomonas converts ammonia into nitrates, which Nitrobacter oxidize nitrates into nitrates. These are the nitrifying bacteria. Okay, these are the autotrophs. The autotrophic bacteria is autotrophs. How many types of autotrophs?
Two types of two types.
Photoautotrophs.
Photosynthetic and
Chemosynthetic.
chemosynthetic.
Photo means light,
Light.
nothing but sunlight. Okay, the source of energy is the autotrophs, light.
Light only.
Chemo means it is a chemical. So the chemical like sulfur, iron, and hydrogen. These are the come under chemical. So it is called as chemosynthetic bacteria. Next, heterotrophs. Heterotrophs. Just wait. Okay. Heterotrophs bacteria. The heterotrophic bacteria are not able to synthesize their own food materials. They obtain the food materials either from living organisms, dead and decaying organisms, in the association with the other living organisms. Right? The photoheterotrophs. In this, these also two types: photoheterotrophs and chemoheterotrophs. Photoheterotrophs. 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 Yes, any idea? Photoheterotrophs.
They will
These
depend upon the green plants for
Mm.
their nutrition.
ఫోన్ చేయరు ఒకసారి అలా అమ్మ ఫోన్ చేయి ఫోన్ చేయి కోసం కానిస్తలేరు ఇగో 5 లక్షలు ఇస్తావు అయ్యమంటుంది గాడ గట్టేనా అలా అంటారు ఆ కుటుంబాలు ఏమంటాడు మీ మామకి ఫోన్ చేస్తాను
Please mute yourself.
అంటే 5 రూపీస్ బాగుందా మీకు ఫోన్ చేయి
Okay. Photoheterotrophs. These bacteria obtain energy from sunlight, but carbon is derived from the organic compound. Example is Heliobacter and purple non-sulfur bacteria. The photoheterotrophs, the example Heliobacter, it is the purple non-sulfur bacteria like Rhodospirillum and Rhodomicrobium. The photoheterotrophs, you know photoheterotrophs, these bacteria obtain energy from sunlight, photo, but the carbon is derived from the organic compound. What is the nature of heterotrophs? Generally, the heterotrophs, they depends on others. So
Others.
the energy source is sunlight, but the carbon is derived from the organic compound. That means that depends the organic compound. So the example, the purple non-sulfur bacteria like Rhodospirillum and Rhodomicrobium. Next, chemo heterotrophs. These bacteria derive both carbon and energy from the organic compounds. They can be classified in the They are classified in the 3 types: parasites, saprophytes, and symbionts. What is the meaning of parasites? Yes, tell me. Please respond.
The parasites means depending the other plants.
Other.
Totally depending.
The parasite means they depend on other living organisms. What are the living organisms? Like plants, animals, and human beings also. Already in the first class, the most commonly our instance in bacteria. What is the instance in bacteria? Commonly the present found in the bacteria. It is E. coli bacteria.
It's disrupt.
It's the most common. So it is the beneficial parasite, and they cause disease in the host, so they are called pathogenic bacteria also. Parasite means they depend on other living organisms. It depends on other living organisms like plants, animals, human beings. They cause disease in the host, so they are called pathogenic bacteria. Example is human disease typhoid. Human disease, for example, human disease typhoid, tuberculosis, pneumonia, cholera, and plague. These are the human disease caused by some parasite bacteria. The disease names is typhoid, tuberculosis, pneumonia, cholera, plague. In plants, cabbage rot, wilt of tomato, cankers in the citrus plants is cankers, and fire blight of beans. These are the some parasites. Next, saprophytes. What is the meaning of saprophytes? It's saprophytes. The
They depend
bacteria.
on dead and decaying matter.
Yes, exactly. The bacteria obtained from their food from decaying organic matter are known as saprophytic bacteria. The examples of bacteria, all the bacillus species are the saprophytes. Okay. The bacteria obtain their food material from decaying organic matter are known as saprophytic bacteria. It is bacillus species. Next, symbiotic. Yes, anyone tell me symbiotic association?
Yeah, symbiotic.
Mutual beneficial.
Mutual beneficial, right? Mutual beneficial. It is the both the organisms are beneficial. It is mutual beneficial. Some bacteria live. Some bacteria live in association with the plants or animals. In this association, both organisms are beneficial. For example, Rhizobium bacteria. The Rhizobium bacteria living on the root nodules of leguminous plants. Yes, you know, Rhizobium bacteria, the leguminous plants. What are the leguminous? What is the role of Rhizobium bacteria?
It stored the nitrous
No, it fixes
Rhizobium bacteria
Nitro-
provide the nitrogen to
nitrogen.
the plant, and plants will give the shelter to the bacteria. It fixes the atmospheric nitrogen into the soil nitrates.
yes, it fixes the atmospheric nitrogen into combined form. Combined form is nitrogenous compounds. Generally, the plants cannot utilize directly nitrogen. How to utilize the plants the nitrogen? Nitrogen is the major and essential elements for growing and development purpose of the all the crop, all the plants. So how to utilize? It is
Root nodules.
the Rhizobium bacteria contains in the root nodules. The root nodules, it is the symbiotic association. The symbiotic association means both the beneficial. Asymbiotic association, asymbiotic. Asymbiotic. The asymbiotic means there is no association, but they fixing the nitrogen, atmospheric nitrogen. So they called asymbiotic nitrogen. Symbiotic nitrogen means symbiotic association, both the organisms is beneficial. Suppose it is the Rhizobium bacteria, it is contained in the root nodules of leguminous plants. What is the leguminous plants? What are the plants? The groundnut one. What is the scientific name of groundnut? Yes, Arachis hypogea is the plant name is Arachis. Groundnut scientific name is Arachis hypogea. It is belongs to the family Leguminaceae family. The most of the all the Leguminaceae families they produce the root nodules. The root nodules contain Rhizobium bacteria. The Rhizobium bacteria it fix the nitrogen, atmospheric nitrogen. What is the definition of nitrogen fixation? Yes. What is the definition of nitrogen fixation? The conversion of molecular nitrogen into
Nitrates.
ammonium ions or nitrogenous compounds like nitrates, and urea also it is the ammonium ion. Urea also it is the ammonium ion. It is called as nitrogen fixation. Nitrogen fixation is how many types of nitrogen fixation? The chemical nitrogen fixation and biological nitrogen fixation. The biological nitrogen fixations occurs, the chemical nitrogen fixation occurs in the, it is in the natural process, it occurs in the nature. The biological nitrogen fixation in the symbiotic and asymbiotic. Bio means biological nitrogen
Living.
fixation. Living organism. The living organisms, the bacteria, these are the conducting of the nitrogen fixing bacteria. So coming to our topic, the bacteria lives in the association with plants or animals. In this association, both organisms are beneficiated. The rhizobium living in the root nodules, the leguminous plants, it fix atmospheric nitrogen into converted to the nitrogenous compounds. The nitrogenous compounds, the nitrites, and ammonia ions.
Ammonia.
And the legume plants observe the combined nitrogen. In return, the legume plant supplies the food materials to the bacteria. In this chapter, the symbiotic association and the biological nitrogen fixation is a very big chapter. We will discuss in the paper 4, I think, physiology paper.
Yes,
Paper
sir.
4 physiology is
Nitrogen
covered under this topic. What is the biological nitrogen fixation? What are the enzymes involved in this nitrogen fixation? This is a very, very important on your exam, the paper 4 also. So these are the today class. Today we will discuss the ultra cell structure of bacteria and nutrition of bacteria. So I inform most probably tomorrow my class is not there. So because some other work, the personal works, I inform to Kumar sir. Kumar sir, they inform to you. And that time, I think Kumar sir take the class tomorrow class 10 to 12. So Kumar sir will inform you. Okay. Nutrition of bacteria and ultra cell structure of
bacteria.
bacteria. Okay. Any doubts regarding this? Yes. Any doubts? Next class we will discuss about reproduction of bacteria.
సార్ అటెండెన్స్ పెట్టమన్నారు మీరు నెంబర్ చెప్పలే ఆ రోజు.
Yes, tell me.
అటెండెన్స్ స్క్రీన్ షాట్ తీయమన్నారు కదా సార్.
Yes. Take the screenshot. My number in your group. So I will send in the chat box. Already someone I sent you attendance. There is no problem. So the class will close within the 10 minutes. Just brief introduction of reproduction of bacteria. Okay. What is the reproduction?
Okay.
Why it is necessary the reproduction? Reproduction. What is the meaning of reproduction? Yes.
New production of new offsprings
New
from the
offspring.
parental cell.
Okay, right. So the bacteria are reproduced by vegetative, asexually, and sexual reproduction. In bacteria, there is no true sexual reproduction in bacteria, right? There is no true sexual reproduction in bacteria. So what is the vegetative reproduction? Any idea? Fragmentation, budding,
Binary
yes.
fission.
That is under asexual reproduction. Okay. Fragmentation, budding is the most common method in the vegetative reproduction. The asexual reproduction means the asexual reproduction is common method of the reproduction in bacteria. It takes place by the binary fission. Binary fission, endospores, cysts, and conidias and buds also. These are the asexual reproduction. And the sexual reproduction is nothing but genetic
Fragmentation and budding.
genetic
Transmission,
genetic
transduction, conjugation.
genetic recombination. What is the meaning of genetic recombination? The exchange of genetic material takes place. It is
To another
called
bacteria.
genetic recombination. Exchange of genetic material. Exchange of genetic material. What we called genetic recombination. Okay, the sexual reproduction in the transformation and conjugation and transduction. Okay, these are the very important on your theory exams. What are the transformation? Meaning of transformation? How the genetic material transformation one cell to another cells, and conjugation. The conjugation also, the cells is contact, the two bacterial cells contact. How transfer the genetic material one cell to another cell, and transduction. The transduction is a very big chapter. The transduction we will see in the life cycle of viruses. Covers under covers. Okay, this is the reproduction of bacteria in the brief. We will discuss in the next class. Okay.
Okay, sir.
Yes. Any doubts?
Important question.
Yes. My number posted on chat box. Just anyone take the attendance screenshots, send to my personal WhatsApp number. Okay. Thank you. Any doubts regarding these two classes? Yes.
Hello, sir.
Yes. Tell.
Semi-conservative materials means, sir?
Semi-conservative material.
Yes, sir.
General conservative means, any idea?
Protected.
Conservative.
Protected.
So, you some of the cell organelles, semi-cell organelles. Semi means
Half.
half. So these, where it is in the semi. Yes. It replicates by the semi-conservative method. Semi-conservative methods. It is the half of the conservative material. They stored in the, generally they store the full materials of some organelles in the stored in the full materials, but semi means is nothing but half. Generally the nuclear material, the reserve food material stored on the cytoplasm, it is the only half. That is called as semi. Yes. Any doubts? Okay. Thank you, all of you. Thank you.
అసలు ఏంటి తెలుసమ్మా మ్యాగీ ఇంకా పాస్తా పాస్తా
చూపించు మ్యాగీ ఎక్కడ దృశ్యత మ్యాగీ కూడా తెచ్చారా
నేను అడిగాను మ్యాగీని మ్యాగీ